18 is the last chapter we're going to get through. Chapter 18, digestion. And what I'm going to do is I'm going to skip the anatomy part right now. And I'm going to skip all the way to, I'm going to try, because the lab tomorrow we're going to do is the digestion lab and the urinary lab. So don't pee before lab, because you got to pee in a cup. Again. So this time you need more than 3 loopfuls. This time you need more. So drink up, buttercup, before lab. I want to skip as much as I can because I want to talk about the physiology of this. Because that's the part, but we're going to talk about whatever. We'll get to whatever. I don't want to not, I want you to know what we're doing in lab tomorrow for the lab, for the experiment. So, one thing, the digestive system is a big long tube, starts at the mouth, ends at the anus. Depending on where you are in the tube, it's a different organ, right? So you eat food in your mouth, you swallow it into your throat, your pharynx, into the esophagus, into the stomach, into the small intestine, and then we get rid of the waste products at the level of the large intestine. The vast majority, 90% of the absorption occurs in the small intestine. Besides the tube, we've also got accessory glands that are going to secrete juices into the tube. Starting at the mouth, we've got salivary glands. As soon as you put food in your mouth, you start to release saliva. We've also got the pancreas, it sits below and behind the stomach. And we've got the liver and the gallbladder. And all of these guys are going to secrete juices into the tube. They're not part of the tube, but they're connected to the tube to secrete their juices through a duct. Jobs of the digestive system. Digestion. Digestion is not chewing. Chewing is breaking your food into tiny components. Digestion means you're going to break the bonds. You're going to break the bonds of the molecules into their tiny molecular components. So that we can do the second job, absorb them. We have to absorb the smallest components that we can. If it's too big, it ain't going to get absorbed and it's going to go out the other end. Motility through the tube, we're going to have lots of motility. We're going to have some mixing type motility and some propelling type motility. And then we're going to also secrete a bunch of fluids, starting with the salivary glands, pancreas, the liver, and the gallbladder. All going to secrete very important juices into the tube. This is completely deranged differently than your book. To me, I'm linear. We're going to start at the top. We're going to work our way down to the end. And we'll talk about what happens in each section as we go. So, let's look at some of our molecules. Remember, these are the biomolecules, your carbohydrates, your proteins, and your fats. That's what you eat. That's what we need to digest so that we can absorb them. And what it means to digest is to break the bonds. So this is a disaccharide, right? It's a sugar that's connected to another sugar. 2 sugars together make up a disaccharide. If it's a polysaccharide, what does that mean? You got a bunch of them in a chain. Our small intestine is where absorption occurs. Our small intestine can only absorb monosaccharides. Can only absorb monosaccharides. So I can't absorb this. If this is lactose, which is a disaccharide, and I don't have the enzyme to break that bond, guess what happens? It goes into your large intestine, and bacteria love lactose. Oh, micro people, don't they? They love lactose. They like to ferment it and make gas. And this is a solute. So then water wants to flow into the lumen. And then what do you get? Diarrhea, bloating, fluffing. Because your lactose intolerant, which means you don't have the enzyme to break that bond anymore. You don't have it. It doesn't work. So don't eat ice cream. Don't eat milk, because that's where lactose is found. That's too big. We can't absorb that. You got to break it. We only have 3 monosaccharides. We have glucose, galactose, and fructose. That's it. There's only 3. You hook them all together in different arrangements, you get all the different sugars. Proteins. Remember that proteins are a carbon-nitrogen backbone. Carbon-nitrogen backbone with amino acids attached. Right? Amino acids attached. They're huge. Proteins are long. Proteins are huge. Many, 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 many hundreds of amino acids long, typically. Ain't no way we're gonna absorb a protein that's got 500 amino acids, right? That's huge. So what we need to do is we need to break bonds. And we're gonna do it in 2 ways. We're either gonna break the backbone in multiple places. That's one way. The other way is to remove the amino acids. Once we do that, then they're small enough that we will be able to absorb them through the small intestine into the bloodstream. Then remember, we've got fats. Fats are that big E structure, right? We've got the glycerol backbone and then the fatty acid tails. This is another big, big molecule. Ain't no way we're going to absorb that. What we're going to do here is we're going to remove the fatty acid tails from the glycerol backbone. The fatty acid tails can actually enter, as well as the glycerol, in a little bit of a different mechanism. So, talked about the structure. Do not worry about the 4 layered walls. You can cross that off. We did that in anatomy. We're not going to do it again. Don't worry about it. You can remember your anatomy from that. Motility through the digestive tract. We've got 2 main types of movement. Peristalsis is the wave-like contractions that push the food along. And segmentation is random squeezing of the tube to mix the juices together with the enzymes so they can all get digested so we can absorb. The migrating motor complex is just a big series of peristaltic contractions. Usually it occurs in the large intestine, but we'll come back and talk about that a little bit later, next week. So peristalsis, here's in the esophagus. You just swallowed your ball of food, and peristalsis is squeezing behind that ball to push it into the next section. So, what we're going to do is we're going to start at the mouth, and we're going to get through, I don't know where we're going to get through, but we're going to start at the mouth. Job of the mouth is to chew. And we put food in our mouth, we chew it up into smaller chunks. But in order to swallow that food, we have to mix it with saliva. So as soon as you think about food, taste food, smell food, your salivary glands start secreting saliva. And what do we find in saliva? Well, we find an enzyme called amylase. Amylase is kind of an umbrella term for different enzymes that digest carbohydrates, sugars. So if you have lactose, you're eating lactose, you have an enzyme called lactase. Remember, all enzymes end in ASE. You have an enzyme called lactase that breaks that bond. If you don't have lactase, then you're lactose intolerant. Sucrose is broken apart by sucrase. Maltose is broken apart by maltase, okay? They're all amylases. And amylase is an enzyme that breaks down carbohydrates. The other thing that we have in our mouth is an enzyme called lipase. What do you suppose that breaks down? Oh, I tell you everything in the chart. Breaks down fats, right. But what's interesting is that lipase can't work in the mouth. It's not acidic enough. Lipase only works once it gets to the stomach. So you swallow the food, lipase is activated by the acid environment of the stomach. Amylase works in the mouth and kind of quits working in the stomach. Stomach destroys amylase, but not lipase. Mastication, fancy term for chewing. Deglutition, fancy word for swallowing. So, saliva, tiny chunks of food, you chew it up, saliva mixes into a slimy ball called a bolus. It's the bolus that you swallow down. The esophagus doesn't do much. Adds a little mucus to the bolus to help the swallowing, to help it go down. Esophagus is just a connector too. Nothing much happens in the esophagus. So, as far as digestion occurs, what is started to be digested in the mouth? Carbohydrates. I did this lab once because I read about it and I thought, oh, that'd be fun to do. And it didn't work very well. But if you take an unsalted saltine, which is kind of an oxymoron, isn't it? An unsalted saltine, a non-salty cracker, which tastes horrible. Tastes like cardboard. What is a cracker made of? Carbohydrate, starch, polysaccharides, right? If you take that cracker and you just chew it up and mix it with the saliva without swallowing it, guess what it's going to start to taste like? Sweet. Because the polysaccharides, stupid man. Polysaccharides are being broken apart by amylase in the saliva, and now we release these individual sugar molecules that have a very subtle sweet flavor. Anyway, I tried this in lab. The problem was nobody could keep it in their mouth long enough to taste it. Yeah, it turns into cracker soup. And anyway, if you're able to do that, and then by the next semester I was going to try it again, but all my crackers went stale, and I just said forget it. So if you ever buy or get some unsalted crackers, you should try it. It will start to taste sweet. All right, the stomach. Remember that the stomach is under the diaphragm. Stomach is full of acid. The stomach is connected to the esophagus by a little sphincter muscle. Keeps the esophagus safe from the acid. The upper portion is the fundus, the main portion is the body, and the end portion is the pylorus. And then there's another sphincter muscle at the connection to the small intestine. Inside the stomach, we have cells that live in these little indentations in between the folds of the stomach. Remember, inside your stomach you've got all these folds called rugae. And in between those, you've got these cellular pits called a gastric gland. And the cells that line that pit, that gastric gland, make mucus. You've got mucus-producing cells. Goblet cells make mucus. They also secrete bicarbonate. What's bicarbonate do? Binds hydrogen. It's a buffer. So the lining of the stomach, the very edge, the pH of the edge of the stomach is close to neutral. And then there's a whole big thick layer of mucus. So it's the lumen of the stomach where all the acid is, then you've got a big layer of mucus, and then right next to the cells here, the pH is about normal. These are not abnormal, I mean, these are not strange cells that can withstand a pH of 2. But the thick layer of mucus and the bicarbonate that's produced here help these cells survive. The acid is in the middle, the mucus protects the lining of the cells, protects the cellular lining. The other cells that we have in the pit are called parietal cells. They make hydrochloric acid. And remember that the acidic pH of the stomach is about 1 to 2. And then we also have cells called G cells. They're going to secrete a hormone called gastrin into the bloodstream. So they're down at the base of the pit. They're down here. And they're going to secrete a hormone into the bloodstream. That's not going to go into the lumen. It's going to go into the blood. And we'll talk about what that does. And the other cell that's produced, or that's found in the pit, is called a chief cell. And chief cells secrete a molecule called pepsinogen, which is converted to pepsin in the acid environment. Pepsin is a protease. What do proteases do? Digest protein. Proteases digest proteins. What's hydrochloric acid do? Denatures proteins, right? So you eat proteins, they're all folded up. Chewing did not unfold them. Chewing just put them into small chunky pieces. That protein gets down into the stomach. What happens to that protein in the acid? It unfolds it so that pepsin can chop it up. If you don't have hydrochloric acid, how good are you going to chop it up? If you can't denature it, you can't chop it. So the job of the hydrochloric acid is to denature the proteins. It doesn't, it destroys pathogens and stuff by denaturing their proteins. It doesn't blow them up like some kind of a grenade or like melt it or anything. It denatures the proteins, and when proteins are denatured, they don't work right. So parietal cells, hydrochloric acid, secreted by the parietal cells. First job, denature proteins so that pepsin can digest. Pepsin. Pepsi means digestion. Pepsi Cola got its name because it's first, they claimed it aided digestion. It doesn't. It was just a ploy to get you to buy it. But that's how it got its name. Pepsi Cola. Oh, it'll help your digestion.
Did they have like a different, they made it differently back then? Or they were just flat out?
So it
Just came up with a fun name.
It wasn't like Coca-Cola or anything?
No. Coca-Cola got its name because it had cocaine in it, you know that, right? Gave you a little lift. Along with the caffeine. So here's pepsinogen. It's inactive till it gets in the environment of the hydrochloric acid, gets converted to pepsin, which then breaks up our proteins. So not only is the hydrochloric acid important for denaturing, it's also important to activate the pepsin. But you gotta have both. So what have we digested so far? Carbohydrates in the mouth, starting the digestion of proteins in the stomach, and what else? That lipase, right, that came from the saliva, that's starting its job where?
In the stomach.
In the stomach. Okay, I'm going to skip that for now. Let's look at the pancreas. Pancreas. Then we're going to come back here on Monday. We'll come back and we'll fill in the blanks that I'm not filling in right now. Pancreas sits below and behind the stomach. it empties its juices into the small intestine through a little hole here, about 4 or 5 inches downstream of the stomach. So here is where the stomach connects. They removed it so we can see it. Where have we talked about the pancreas before?
With hormones.
Endocrine system. Remember we said only 1% of the cells are important in the endocrine system. The bulk of the pancreas is digestive. 99% of the cells are important in digestion. So what happens is the pancreas makes all digestive enzymes. Pancreas makes amylases, proteases, and lipases. All of them. Pancreas is really important in digestion. And all of those enzymes are going to be shuttled into this little duct. And after you eat a meal, those enzymes will be released into the small intestine. Because what's going to enter the small intestine from your stomach? Food. So the pancreas releases its juices because of a hormone called CCK, which, hang on to that, we'll come back to that. So what's released by the pancreas? Some bicarbonate. Bicarbonate's a buffer. Remember, what do you suppose the pH of the juice is coming from the stomach here?
1 or 2.
It's pretty high. I mean, you mix it with some saliva and you mix it with some food and the pH rises a little bit, but it's still pretty acidic. And this duodenum doesn't want to be acidic. So we're going to release some bicarbonate, we're going to neutralize the acid, and then the pancreas is also going to secrete amylases, proteases, and lipases. So we're going to really digest in the small intestine. Okay? Skipping the rest here, I'm going to go to the liver. The liver stores and concentrates. The liver makes a substance called bile. And bile is stored and concentrated in the gallbladder. The gallbladder is literally green. Bile is kind of a greenish, ugly green color. And what we have in bile is comprised of cholesterol, water, my board's getting messy, bilirubin, which is a breakdown product of hemoglobin and red blood cells, right? It gives the poop its brown color. It's very concentrated in the poop. And something called bile salts. It's the bile salts here that are the important component. Bile salts come from bile, and what they do is emulsify fats. Remember, all these juices that you're secreting in your digestive tract are water-based. What happens when water and fat mix? They don't. So when you eat fatty meals and it mixes with all the water stays here, the fat all comes together, right? Fat likes fat, water likes water, so you get these big fat balls. And what bile salts do is it breaks those big fat balls into little fat balls. Emulsify means it's like soap. Soap emulsifies fat. It breaks the fatty components from a big layer to tiny layers so that it can be washed away, like on your hands. So it's going to take these big fat balls that are mixed with all the water and break them into little ones. Because if you have a big fat ball and you've got all these fats in here, our big E structures, in my big fat ball, how do my lipases, they can't get to the middle guys, right? They might be able to digest these fatty acid tails off, but what are you going to do to the big fats in the middle? You can't. So if you break them into smaller components, they're more accessible for the lipases to do their job. If you've got gallstones and you can't get bile into your small intestine, one of the symptoms is you have greasy, oily poop because you didn't digest the fats and absorb them and you pooped them out. So that's what the main job of the liver, as far as digestion in this case, is to break the big fat balls into little fat balls so that our lipases can get to them and remove the fatty acid tails.
Are
And
gall,
that should be good enough to get through lab.
are gallstones like